So, dear students, today I'm going to discuss on the topic microbial structure, isolation, and culture. Since the topic is very vast and it's a combination of three topics. One is microbial structure, other is isolation and culture. But the reason for taking all these three is very simple. We have collected questions which are generally asked in the examinations, competitive examinations, and we found that if this can be synchronized and presented in the form of capsule, so that can be better for your competitive exam preparations. So one by one, I'm going to start all the topics. And obviously, along with that, I'm just going to share some questions also. The pattern of the questions that is very important for you to correlate the theory as well as questions. The first is in the structure. I'm going to start with the bacteria because most of the question asked from the bacteria. Structure of bacteria. It was first observed by Antony von Leeuwenhoek in 1676. And in bacteria, you can find three categories. It means in the structure of the bacteria, there are three levels. The first is internal structure, that is cytoplasmic content, which also contain nucleoid. Nucleoid is also known as bacterial chromosome, which is a chromosome without any nuclear membrane. So we are saying it is the difference between the nucleus and nucleoid. In bacteria, there is no nuclear membrane present. Then plasmid, which is also known as extra chromosomal DNA present in the bacteria. Some bacteria, ribosomes are different than the eukaryotes. They have 70S type of ribosome, and many storage granules are also present, which contain carbohydrate, protein, lipid, and sometimes pigments also. The next part is the cell envelope of the bacteria. The cell envelope contain two or three layers. In gram-positive bacteria, it is two layer. Of course, inner is cell membrane and the outer next one is cell wall, which is made up of peptidoglycan. If it is gram-negative bacteria, you will found three layers. Inner one is obviously, it is cell membrane. The next is cell wall, which is thin layer of peptidoglycan. And the third, that is outermost layer, is the lipid membrane layer. Then the third stage is, or categories are basically external structure. In different bacteria, different appendages are present as an external structure. Like some possesses flagella. Flagella is responsible for the movement. Then some possesses fimbriae, which is important for the adhesion of bacteria. With the help of fimbriae, the bacteria is able to adhere on a particular surface, and it is very important for their colonization. It is very important for the disease spread by them. It means that most of them are virulent also. And sex pilus, this is the third appendages. Those bacteria which possesses sex pilus, they are able to transfer their genetic material from one bacteria to the other one. The next is glycocalyx. It's a outermost layer in some of the bacteria which make them slimy. You may heard about the previously heard about certain experiments in 12th also that is related to the transformation, in which you observe that glycocalyx was present, and that type of bacteria was known as virulent type of bacteria. So glycocalyx is providing basically virulency to the bacteria. So these are the overall structure of the bacteria. You can see it in different way also, like here you can see the outermost layer. Of course, it will be either pili or it is flagella, etc. The envelope is made up of basically the outer is slime layer, the next is slime layer, that is the capsule, the next is cell wall, and the third is plasma membrane. The inner one is cytoplasm, which may contain plasmids, but all bacteria contain nucleoids, etc. Ribosome are 70S type as well as food granules. So this is the structure of bacteria. One thing I want to mention here that they are not possessing any specific organelle, like very specific double membrane bound organelle like mitochondria, like uh, uh, endoplasmic reticulum. But it is present in eukaryote but absent in the prokaryote. So how bacteria is managing all their metabolism in absence of all these things? So let me tell you, the cell membrane of the bacteria is very important. The cell membrane move inwards and making a loop-like structure, and this is called mesosomes. Mesosomes are important in the sense that they are very, very equivalent to mitochondria. These mesosomes are extension of cell membrane. which possesses enzymes responsible for ATP synthesis. So mesosomes are equivalent to mitochondria. Second thing is, if I'm talking regarding photosynthetic bacteria, so all the photosynthetic pigments are also present on the membrane of the bacteria. Then third is talking regarding the function of protein folding, etc., or protein formation. So everything is going to happen in the cytoplasm only. So these things are happening in cytoplasm. This is how bacteria is very simple and able to manage all these important functions using their available structure like cell membrane and in the cytoplasm also. Then coming to the next part, it is the shape of the bacteria. The shape of bacteria is are various type. Suppose I am talking around the round or oval shape or circular type. It is known as cocci. Cocci may be joined together to make diplococci. And one of the best example is Streptococcus pneumoniae. Then next is Staphylococci. It means the bunch of the bacteria. The best example is Staphylococcus aureus. Directly questions can be asked with the examination. So in the exam related to the examples. So what you should do is always you should remember some of the example like Staphylococcus aureus, then Streptococcus pneumoniae. So these are important examples which I mentioned here. Then Streptococci, which is a linear form and made up of several number of this round cocci. Actually, it's a combination it making a straight chain. The example is a Streptococcus pyogenes. Then they can make tetrad or octadic structure also. The tetrad and octad, you can see octad structure you can find in the Sarcina. If you talk regarding the bacillus, these are basically the shape is different than the cocci. You can easily able to find this rod shaped, and if these rod are joined together, it's some they are making chain, and it is called chain of bacilli, which is very common in the bacillus group like Bacillus anthracis. Some of them may possesses flagella also. So flagellated rods are also present. Example is Salmonella typhi. Then some of them are spore former. One of the best example is Clostridium botulinum. They are able to produce spores and they are toxic also. You know very well they are able to produce toxins. So Clostridium botulinum is associated with the toxin productions. If you talk regarding the other group of bacteria other than this cocci and bacillus, some are comma shaped like Vibrio. Example, of course, it is very common Vibrio cholerae, disease causing bacteria. Then Spirilla. One of the best example of Spirilla is Helicobacter pylori, which is causing gastrointestinal disorder. It is present. It is sometimes present in the human stomach. So they are able to survive in the stomach. Actually, they are present in the pyloric region, and hence it is called Helicobacter pylori. And the next one is spirochete. They are also a spiral in structure, but much more longer than the Spirilla. Best example is Treponema pallidum. Next, you can see some examples of the question. The cocci, which form a bunch and regular patterns, are obviously everyone know to this answer. I think everyone knows it is diplococci. I'm not giving so much stress on this question. Reason is if I will go through all the questions, I cannot complete in the given stipulated time period. So some of the questions I may ask, but some questions I'm just going to solve by myself also. So the answer obviously it will be diplococci. The next is it's a bunch. It is a staphylococci. This is this is analogous to mesosome of the bacteria. So if I'm saying about the mesosomes, just now I explained this already. So mesosomes are equivalent to mitochondria. Now come to the cell wall. The cell wall of the bacteria. Why I'm choosing cell wall is because so many times questions are asking specifically from the cell wall. The cell wall of the bacteria possesses peptidoglycan, which is also known as murine. It is basically made up of polysaccharide chain cross-linked by the peptide containing D-amino acid. Yes, we should remember here it is D-amino acid. The peptidoglycan is itself made up of N-acetylglucosamine and N-acetylmuramic acid. NAM-NAC crosslinks are present. And here you can also see teichoic acid and lipoteichoic acid. Then some specific terminology related to the cell wall is the bacteria which do not possess a cell wall. It means, I'm not saying it is not possesses, but if you remove cell wall of the bacteria, it is called protoplast. And if you entirely removed, it is protoplast. And if you are partially removing the cell wall, it is called spheroplast. And one important point is in the mycoplasma, the cell wall structure is absent. So there is a difference between absence and entirely removed. If it is removed, it is called protoplast. And in mycoplasma, it is naturally absent. You can see the structure of the cell wall. This is gram-positive bacteria having two layers. The outer layer, this is basically peptidoglycan, and the next layer it is cell membrane, which is selectively permeable. Whenever I'm talking regarding membrane, it means I'm talking about the permeability also. It's a specific feature of the plasma membrane. Besides these two things, they are also possessing teichoic acid as well as lipoteichoic acid. So this is the two-layer structure of the gram-positive bacteria. But simultaneously, if we'll talk regarding the gram-negative bacteria, they have three layers of the structure. Two obviously present the same one, that is the cell membrane is present here also, and peptidoglycan also it is present. But you can see here it is thick, and here it is very, very thin compared to the gram-positive bacteria. And additional layer, that is third layer, is outer membrane layer. This outer membrane is basically made up of lipopolysaccharide, phospholipid. They also contain lipoproteins. So the third layer is making it different. And due to the third layer, that is outer membrane layer, this gram-negative bacteria is having a specific property that they are able to resistant from so many antibiotic. So if I'm talking regarding antibiotic resistance cases, gram-negative bacteria are more resistant to antibiotic as compared to the gram-positive bacteria. Reason is very simple. In gram-negative bacteria, extra layer, that is the outer membrane layer, is present, which they make them more resistant towards antibiotic. Okay. Now moving to the next part, same structure, that is regarding the gram-negative bacterial cell wall. If I'm going to focus, you can see here that as I explained this structure about the outer membrane, this outer membrane is basically made up of O, polysaccharide, and lipid A. So basically this slide I'm showing because I want to show the structure of LPS, lipoprotein, and surface protein. The outer membrane is made up of all these things, but the lipopolysaccharides are again, these lipopolysaccharides are again made up of two things. One is lipid A and second is O polysaccharides. So that you should remember, that is the additional point. Besides that, you should know about the S layer of the bacteria, that is the surface layer. Surface layer is basically a crystalline layer on the top of either gram-positive bacteria or gram-negative bacterial bugs. Why I'm saying bug? Because they are disease-causing of bacteria. So in most of the disease-causing bacteria, you can find S layer, that is surface layer. And this S layer, of course, if it is present, it is providing virulency. So most of these virulent bacteria are having S layer that help in the adhesion. And if it is adhesive, of course, they may cause disease. Then come to the next one. Again, some questions are asked on the basis of that cell wall. One of the questions asked in the JNU in 2017 is, with reference of the bacterial cell wall, which of the following statement is correct? So this question you may solve, but I'm also solving parallelly. That the gram-positive bacteria lacks peptidoglycan layer. This is wrong. Of course, the peptidoglycan layer is present in both. So A and B is wrong. C is saying that both gram-positive and gram-negative bacteria possesses peptidoglycan and outer membrane. This is wrong because you know very well that outer membrane is not present in the gram-positive bacteria, and hence you will come to the fourth number where it is saying that both gram-positive and negative bacteria possesses peptidoglycan, but it lacks in the gram-positive. So answer will be the D number. Now, which of this is exposed to the outer surface of the gram-negative bacteria? Answer is well known to you. It is LPS layer. Similar type of question asked in the GNU again, that bacteria depends on the permeability barrier of. So whenever saying about the permeability, you just close your eye and you can write down the answer, that is cell membrane, because permeability is associated with the cell membrane. Now next question is, which of these bacteria is resistant to the penicillin as it lacks cell wall? So now it will make confusion, but you know very well that cell wall is lacking. It means the answer is obviously mycoplasma. Okay. So by this way, you are able to solve this type of answer which is related to the cell wall. Now come to the main core part, that is flagella. See, flagella again, if you'll see, I'm just going to emphasize the major points that you should remember is flagella are present in both gram positive and gram negative. But there is a difference in the structure of the flagella. In gram positive and gram negative, the number of rings are differed. So in gram negative, you can find that L ring is present in the outer membrane, that is L. The next is P ring. P here, P is related to the peptidoglycan. In peptidoglycan, you can find P ring. And in the plasma membrane, there are two rings, S and M. So in the order, you should remember LPSM, LPSM. So L is outer, P is peptidoglycan, S and M is present on plasma membrane. This is regarding the gram negative. In gram positive bacteria, you will find only two type of ring. One is S ring and M ring. It is absent. It is not showing any L ring and P ring in the case of gram positive bacteria. Okay. Now moving towards again questions. L ring is in gram negative bacteria is associated with. So this answer also it is easy to understand. Just now we observed that L is part of gram negative present in outer membrane, and flagella in bacteria is enable them too. The flagella is associated with the, I just said it is
Locomotive.
yeah, very good, locomotion. Now, another type of questions may be asked from the archaea and bacteria. See, I'm not going to talk regarding any regular lectures and not going to talk regarding the structure of bacteria in detail. Only those portions I mentioned, which is actually as in the exam. So I just left the archaea as well as other part of the bacteria. I'm just going to show the comparative study. And again, in comparative study, there are n number of points, but you should remember the 4 points mainly. The cell membrane, the first one. The bacteria possesses ester link in their, which is basically, it is a lipid layer. Of course, it is cell membrane, lipid possesses ester. But in archaea, the ester is not present. Instead of that, you can find ether. So ether link lipid will be present. If coming to the next part, that is cell wall, the difference between these two is in the cell wall, you can find that peptidoglycan is present along with the other things also. But in the archaea, there is no peptidoglycan, no murine. It is pseudopeptidoglycan. So remember this point. In archaea, peptidoglycan is absent and it is possessing pseudopeptidoglycan. These 2 points are actually important. And if I'm talking regarding the gene structure, so bacteria is having unique gene structure in the sense transcription and translation. They have unique transcription and translation that you have studied about the bacterial or prokaryotic transcription and translation. But archaea are matching with the eukaryotes in terms of their transcription and translation. So this is not unique actually. Archaeal transcription and translation is very much equivalent to the eukaryotes. This is the third major difference. And fourth difference you can see is, of course, this is the part of gene related, but here I'm mentioning separately because questions are asking separately that in the bacteria you can find formyl methionine, whereas in archaea you can found methionine as the first amino acid. That is the initiation. Okay, so these points you should remember. Now again, one question is asked in the GNU from the same point that the cell wall of the archaebacteria does not contain. क्या नहीं होता है? So just now I said that अगर आप बात करोगे archaea की, of course protein is present, polysaccharide is present, and pseudopeptidoglycan, it means pseudomurine is present, which is not present. Answer is known. It is peptidoglycan. And methanogen, it's a very common questions. Methanogen belongs to, that you should remember, it is belonging to the archaea, archaebacteria. So after bacteria, I'm leaving bacteria and coming to the next part, that is fungi. Some of the questions may ask from the fungi also. The major point that you should remember is eukaryotic. Fungi are eukaryotic, non-photosynthetic, and examples of them are mold, mushroom, which are multicellular. But yeast is a fungi which is coming under the lower fungi. It is unicellular. And the cell wall of the fungi contain chitin and they store glycogen. okay, as a energy source. Then these fungi are saprophytic in nature, and they are having hyphae-like structure. And this hyphae, the single one is called hyphae, if I am talking regarding one, and the network of hyphae is called mycelia. The study of fungi is called mycology, and when they are going to make asexual spore, on that time the hyphae is modified, and after modification they are making fruiting bodies. This structure also you should remember, these fungi are showing root-like structure that is known as rhizoid, and they have a horizontal hyphae, and this horizontal hyphae is known as stolon. And this vertical one is known as sporangiophore, and this sporangiophore contain sporangium. So this structure also you should remember. And some of the general characteristics, again, important is type of spores. Here you can find some questions asking from the spore. See, fungi produces two kind of the spore. One is asexual and second is sexual spores. Asexual spores are arthrospore, chlamydospore, sporangiospore, conidiospore, zoospore, and blastospore. You should remember the name of these spores. And sexual spores which is produced are asco, basidio, zygo, and oospores. So there is a difference between the spores of asexual as well as sexual mode of reproduction. Okay. And one more thing is fungi is going towards asexual reproduction only when the condition is favorable. And when condition is unfavorable, they move towards sexual reproduction. And they are producing different kind of spore in different conditions. Then three examples you should remember. All the aspergillus are producing conidiospore. Rhizopus is able to produce two kind of the spore, sporangiospore as well as zygospore. And club fungi is producing basidiospores. So these are the example that you should remember. Each and every point is important. Dear students, I am not going to teach microbiology. That is not possible to complete in 1 hour along with the questions. Only mentioning point is it's like a capsule, and important point I mentioned. Always try to remember all these points. So that DAS produces basidiospore. Which one is producing basidiospore? Just now we have gone through that PPT. It is club fungi. And other thing is which of these spores are characteristics of black braid mold, that is Rhizopus. And we have gone through two kind of the spores. One is sporangio and second is zygo. So obviously the answer will be sporangiospore and zygospore. So D number part. This is how they are asking questions. So that's why I am including all the question patterns. And some of these are asked in the multiple times in different type of examination. So only I mentioned JNU if it is asked from the JNU. But in the other competitive examination, whatever the questions are asked, I have not mentioned here because it is a mixed type. Some asked in the JAM, some asked in other examinations like BHU, some examine direct life science of JNU. So like in that way, but I have not mentioned the names. Now coming to the next part, that is algae. Study of algae is known as phycology, and they are having many inclusion bodies like starch granules. They have oil droplets, vacuoles, etc. These are inclusion bodies present in the algae. They have thallus-like organization, as you know very well. Thallophyta, they're considered as thallophyta. Okay, so they have no any cytoskeleton-like organization. That's why it is called thallus organization. Not cytoskeleton, I should say that rigid structure are not present, that vascular system is not present in the algae. They have thallus organization. I should not say cytoskeleton, I should say about the vascular system is absent in the algae. The cell is eukaryotic, obviously, and the cell wall is made up of cellulose. And besides that, some other substances are also deposited like pectin, algine, chitin, fucoidine, etc. The pigment they are able to produce are chlorophyll A, chlorophyll B, xanthophyll, carotene, phycobilin, and phycobiloproteins. So these are the different type of pigments produced by them. Some points which is important for the algae is a red tide. Many times question asked from the red tide. Who is producing red tide? It is dinoflagellates which is causing red tide. Okay, and this is actually red tide is related to the dinoflagellate and it is toxic. One of the questions many times asked about heterocyst formation. Heterocyst formation is related to the cyanobacteria. There is always a question that where I have to mention cyanobacteria because it is a bacteria, but earlier it was classified under algae. So anywhere if you want, you can keep anywhere. So I'm just keeping it here, cyanobacteria, or it is also known as brown green algae. It possesses heterocyst, and this heterocyst is actually it's a specific type of cell structure present in this cyanobacteria and responsible for nitrogen fixation. So direct questions can be asked from the heterocyst, which is related to the or associated with the nitrogen fixation. Again, the name are sometimes mentioned that algae are classified on the basis of different pigments they possesses. Like green algae is also known as chlorophyce. Chlorophyll pigments are present in them in the abundant amount. Like in the same way, brown algae is also known as phaeophyce and red algae, that is rhodophyce. Then next, coming to some questions may ask from the algae is characteristics used to place algae into divisions include all of the following except. So what type of characteristics are actually used to place algae? So see, they form of storage material. You know very well all the different type of inclusions are present over here. Then flagellar number and locations. Of course, algae is possessing this. Accessory pigments, these are also present. So answer will be all of the above. Characteristic used to place algae into division include all of the following except. Okay. This is a little bit tricky question. The dash, oh, sorry, I have already given answer. The dash is the vegetative body of algae. Answer is thallus. Now coming to the protozoa. Protozoa, this is the fourth part of microbial structure. Protozoa, as such, they are unicellular, and earlier they was classified in the animal kingdom, and the unicellular animals was placed under protozoa. They are eukaryotic, of course, protista, and since they are originated from animal kingdom, obviously, or animal origin, they do not possess any cell wall, and their mode of nutrition is also holozoic. One important question asked from the protozoa is the freshwater protozoa have contractile vacuoles that is responsible for the osmolarity. They pump out excess of water using this contractile vacuole. So many times I have seen this direct questions are asking from contractile vacuoles. Then next function of this protozoa is some of them may be photosynthetic or absorptive, ingestive, or they are playing important role in the recycling of the nutrient and hence part of the decomposition process. Okay, so they are heterotrophs, and some of them are pathogenic if access nutrient from the living organism. It means there are many protozoa which are actually parasitic in nature and hence they are pathogenic, like amoeba causing amoeboisis, like in the same way trypanosoma, giardia, coccidia. There are many protozoa which are able to cause disease. Okay, you heard about all this. Malaria, it is plasmodium, giardiasis, giardia, etc. CC fly, trypanosoma gambinis, etc. So which of the following is least likely to have a rigid cell wall? I think rigid cell wall, you should understand it. It is protozoan, they don't have cell wall. And protozoa have several type of vacuole. The function of contractile vacuole is, just now I mentioned. If I'm saying vacuole function, of course, it is osmotic balance. So they maintain osmotic balance by continuous water expulsion. Now coming to the fifth structure, that is the last structure of the microbiological structure, virus, which is submicroscopic particle, and they have any one type of nucleic acid, either it's DNA or RNA. They don't possess specific enzymes and hence they are not able to replicate separately. They have to insert their genome into their host and hence they are not able to replicate separately. They can replicate only by utilizing the machinery system of their host. They may be DNA, maybe RNA, but can't both. So questions are asked from this example also. So viruses may be DNA virus or maybe RNA virus. The questions are always asking that if it is RNA virus, then how they are replicating. So again, RNA are two types. RNA virus, they may be positive sense or they may be negative sense. Okay, so there is a separate part of that positive and negative sense, but most of them possesses a reverse transcriptase enzyme, which make RNA which make DNA from RNA. They are incapable of metabolism, obviously, and replicate only inside the living cell. In this structure, you can find that there is a protein code. The nucleic acid is surrounded by protein code. So nucleic acid is present in the center, and protein code is basically this capsid layer. And the outermost layer is also present in some of the virus, not all, some of the virus. When this virus, some of the virus may enter into the host, and uh, when they exit out or coming out from the host, they are taking certain part of that plasma membrane of the host only, and hence they are known as enveloped virus. They are actually more virulent, more pathogenic. So they are virulent type and easily able to enter inside the host cell. And these are called enveloped virus. So again, virus are of two types, enveloped or non-enveloped virus. But whether it is enveloped or non-enveloped, they must possess genetic material, either DNA or RNA, as well as capsid. So viruses have different shapes. You can find different shape. Two shape you should remember at least. One is polyhedral shape, which are commonly present in the adenovirus, and helical, which is present in tobacco mosaic virus. The life cycle of virus, if you'll see, then the first step of this virus life cycle is adsorption. They are always adsorb upon their surf on the host. Say, for example, I am saying that this is the host cell. Once virus attack on the host cell, first they have to follow the pattern of adsorption. So once they adsorb, remember it is adsorb. Okay, so adsorb. Then next is penetration, where they penetrate. It means they inject their nucleic acid, whether it is DNA or RNA. They inject their nucleic acid inside the cytoplasm of the host, and this capsid will be remain left outside only. Capsid is not entering inside most of the time. Okay, the next one is latent phase. Latent phase is the period where this nucleic acid, that is viral nucleic acid, is going to utilize the content of the cytoplasm, and hence they are able to replicate themselves. They are utilizing enzymes of the cytoplasm, and hence and other things also, like sugar base, phosphate, etc., and they are able to replicate and increase their genome, whether it is RNA or DNA. And then they can pack themselves. Again, from here they can find two types: either they are in the lysogenic or lytic. So if they are following lytic, it means they will be lyse. They will break down this that membrane, and hence they will be lyse out. So you can see that one virus is entered into the host, and they are not directly entering, but they're entering their nucleic acid. They are able to replicate, they increase in their number, they break down the membrane, and they are remove them out. This is how the one virus can make several copies inside the host cell. Now, viruses are again of two types. It may be active virus, or it may be hidden virus. First, I'm talking regarding the active. The active virus, once virus will attack on the host, what they will do is, as I said, that it will adsorb, then they penetrate their genetic material, and this genetic material may bind with the genome of the host. Okay. I'm saying about active, right? Okay. Inactive, they are not going to bind with the genome. They insert their genome. Now their genome will increase in their number. It means viral genome is increasing. The copy number of this viral genome will increase. And not only that, of course, they are able to make their membrane, their capsid also. And then they can just break down the genome of the host. So host genome will be broken down into several fragments, and then this virus will be move out. So they will be exit out, as I just mentioned, that they will be following this lysis pattern, the lyse and removed. So they are very quick. They are not retaining in the cell for any longer time period, and hence they are known as active. Active because very active. Once they enter into cell, replicate, and after replication, they just pack everything and then they left out. By this way, they can spread very fast. And some of the virus, you know very well, they are very active, like our this COVID-19. They're very active. They're taking very less time for the spread, and many viruses are spread within 1 to 2 days only, and they can spread with a faster rate. But some viruses are hidden virus. like rabies virus. They can stay for the longer time period. You may heard that the dog or some animal bites some person, and and he got disease after 10 years or 15 years. So that is the hidden, because the hidden virus may stay for the longer time periods. So hidden virus are those virus which are inserting in the same way that job insert their genome, but their genome, viral genome, will integrate with the host genome, and after the insertion, they will be remain present in the lysogeny state for the longer time period. But whenever the time will permit, it means the condition is favorable. Again, they enter into lytic phase. It means same in the same way they can increase their copy number, the pack, they break down this genome of the host, and they move out. So this is the case of the hidden virus. Now, one question asked in the exam, that which of the following is a false statement? This is the fifth or sixth question from the JNU, and the answer, I don't have time. If you want, you can. False statement is archaebacteria mostly grow in the harsh condition. Of course, it is true. Several bacteria are pathogenic. Of course, virus are cellular. No, virus is considered as acellular because they are between the living and dead. And answer will be C number. The next is viral nucleocapsid is a combination of. So nucleocapsid, as you know, it's a combination of course, genome and capsid. So that's why nucleo and capsid. Nucleo is indicating genome and capsid, it is protein part. And next question is, in order of for a virus to replicate, what they have to do. So if the virus has to replicate, they have to insert their genome inside the cytoplasm. So the answer should be C number. Okay. Reverse transcriptase is a useful enzyme to have when. So what is the role of reverse transcriptase? In RNA virus, they have important role which convert RNA to DNA. So first number will be the answer. And the first step in infection of a host bacterial cell by a phage is the first follow adsorption, not absorption. So answer will be the first one. Okay. So that I'm not giving you time to solve because I have mentioned n number of the questions and it will not complete in the stipulated time period. So coming to the isolation of bacteria, next part. See, isolation mainly refers to the separation of a strain from natural or mixed populations. Okay, from the environment. So you can isolate from the mixed population. You can isolate from any environmental sample like soil, water. You can isolate from the skin, flora, oral flora, from infected area. Anywhere if it is present, you can isolate. So isolation can be done by several methods like spread plate, pour plate, streak plate, micromanipulatory method. Again, for the pour plate, before pour plate, you can go for the serial dilution or loop dilution methods you can follow. So this is spread plate, or first you should generally, we are going for the serial dilution because the number of bacteria is more in the given sample. And to dilute the number of microorganisms, we should subsequently dilute it. Subsequent dilution is called serial dilution, in which you can take the sample. If it is liquid sample, you can directly take it. If it is solid, you know very well that you have to make suspension, like 1 gram soil dissolved in 9 ml distilled water. In that way, you can make suspension also and pass 1 ml in the first one. Of course, it will be -1, then continuously pass 1 and the dilution will be -1 to -6. And then you can just spread as per your need. Of course, the number of micros will be continuously decreasing. The only information that you can get from serial dilution is the number of bacteria is continuously decreasing using this technique. After serial dilution, you can go for either spread plate or pour plate technique. So spread plate is saying that you can take this some little amount of the sample, like microbial sample or environmental sample, like 100 microliter you can take and pour over the plate, and then you can spread using the spreader, glass or use and throw plastic, then incubate. And after incubation, next day you can get a typical bacterial colony which are separated from each other. Like in the same way in pour plate method, you can first pour the sample and then pour upon that, you can pour 20 ml media and they can incubate and you can get the separate isolated colonies. Or pour plate can be done by the other way also. You can take original bacterial sample and be mixed into the agar, that is 20 ml nutrient agar, and then you can pour over the plate And in that, you can add microbial sample also. So bacterial suspension mixed with the molten nutrient agar when it becomes coolant, and then you can pour, and you will get the spreaded colony. So this is the method for the pour plate method. Or otherwise you can follow strict plate. Strict plate method is for streaking by different ways, but the actual benefit of using strict plate method is here you can get isolated or pure culture. So that is the beauty of the strict plate method, in which you are getting pure. Okay. Then another method which is not well known to everyone and not mentioned in everywhere, but it is important nowadays. This technique is advanced, and many times again some advanced questions are asked by the examiners also. Micro manipulators. Micro manipulator is an instrument which are used to choose or pick out a single cell or single colony. So from the suspension itself, like hanging drop, you can use, and from the suspension only, you can see through. You can go through the microscope, and you can select any one colony with the microscopic observation. You can select. You can see different colony, any colony which you want to take it out. You can take pick, and after that you can spread, or you can pass to the other plate. So this is the micro manipulatory method, but the only thing is it is expensive. Instrument is expensive, and it requires expertise that you have to balance for the microscope, and simultaneously you have to pick colonies. Okay. But questions are asked. You can see isolation of pure culture refers to. So there is a confusion always that whether what will be the answer. But answer is very clear. If you want to isolate, it means you have to separate a single colony. And next question is related to, of course, micromanipulator. Which device is used to pick a single bacterial cell from a mixed culture? Answer is micromanipulator. And that's why I just mentioned this point in front of you. Now, the third point, which are coming to the third point of the day, that is bacterial growth. This is our third topic. And the last topic, you can say bacterial growth. See, bacterial growth, it means basically proliferating of bacteria. If I'm saying only about the growth, it means it includes increase in number and increase in biomass. If I'm talking regarding any mycelial type of organism like fungi, then biomass is increasing. But if I'm talking regarding the bacteria, basically we are concerning with the proliferation of bacteria by increase in number. That is, the growth is related to mainly increase in number. Of course, after increasing number, they have to basically grow till they have to reach to their mother size. But basically it is related to the increase in number. So it is associated with the growth is in bacteria associated with increase in number. And of course, for every division, they have to follow the cell replication step. For the replication, of course, the DNA will be copied, and this DNA will be separate from each other. And then the cell divide. After division, two daughter cell will be produced, which again reverse. It means they increase in size and they follow this pattern. I have seen that many questions are asking from this carbon source, energy source, as well as electron source. So what I did is I put some of the part here also, because though it is not directly linked to the growth, but it is said that the growth is not possible without the carbon, energy, and electron. So along with the growth, you should remember about some certain points regarding the classification of bacteria according to their requirements, nutritional requirements. So bacteria require three important things: carbon, energy, or electron. So regarding the carbon source, if bacteria is solely depend on the CO2 as a principal carbon source, they are known as autotrophs. And if they are utilizing reduced or perform organic molecules instead of carbon dioxide, if they are utilizing this organic molecule, they are known as heterotrophs. Like in the same way as an energy source, if they are dependent on light for the energy source, it is called phototroph. Or if they are dependent on the same organic molecule, they are not dependent on light, they are known as chemotrophs. And regarding the electron sources, if they are dependent on inorganic molecule, it is called lithotrophs. And if it is organic molecule, it is organotrophs. So you can see organic molecules is carbon source, electron source, this energy source, as well as electron sources. Okay. So on that ground, you can say the name autotroph, heterotroph, phototroph, chemotroph, lithotroph, and organotroph. But combinedly, if you will study it, then it will be look like this one. Some bacteria, the bacteria may be utilizing carbons, utilizing light as an energy source. So source of energy is light, so it is called photo. If electron they are utilizing, a source of electron is the source of electron is inorganic, that is litho, and carbon source is CO2, that it is auto. So photo, litho, auto, photolitho, auto, or it is also known as photoautotrophs. So in this way, if only first 3 points are mentioned, you are able to solve the answer. Or if these answers are given, you can write this one. Like in the same way, another example is photoorganotrophic heterotrophy. So photo, it means light. Organo, its source of electron is organic, and it is hetero. The carbon source is not carbon dioxide. Mostly it is organic. Some exceptions might be present in some cases, but mostly it is organic. So chemolithotroph, if I am saying chemo is a chemical energy source, litho is an inorganic electron source, and it is auto, it is CO2 as a carbon source. And best example is sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria. See, example is very important, huh? Because sulfur, hydrogen, nitrifying, iron. I am mentioning this slide because questions are directly asked from here. So I don't want to miss these slides. Okay. And organotrophic heterotrophs, of course, it is chemo means chemical energy source. Organic, it is electron, and hetero, it is organic carbon, it is hetero. Okay. So example are protozoan, fungi, etc. I just I can say you that you should remember these three. Photolitho, the best example is this purple and green sulfur, and cyanobacteria. That you should actually remember. Purple and green sulfur bacteria is the example of photolithoautotrophs. Purple and green non-sulfur. This is purple and green sulfur. And if it is non-sulfur, it is coming under the photo or organoheterotrophs. So this is photo. It means these are actually photosynthetic bacteria. So purple and green sulfur bacteria are photosynthetic. Purple and green non-sulfur are also photosynthetic, but there is a difference. One is lithotrophic, other is organotrophic. So this is the basic difference between these two kinds of the bacteria. Whereas the similar type of bacteria, there's a hydrogen bacteria or iron sulfide oxidizing bacteria or nitrifying. These are the good example of chemolithotrophic bacteria. So at least these names you should remember, which are commonly asked in the exam. Now come to the bacterial growth. Bacterial growth, it can be expressed in terms of culture. So there are different type of culture operation. Generally, we are following batch culture. In the batch culture, of course, the amount of the media is fixed. The volume is fixed initially, and up to the last only that we are working with that volume only. So it is fixed volume of nutrient is provided here. And you can get the initial phase, lag phase, where bacteria is trying to adjust with the environment. And once they are adjusted, they try to grow with a faster rate because there is no any hurdles for the uptake of nutrient. So once they are taking nutrient, they are growing, and they are growing with the maximum specific growth rate, and hence it is known as exponential growth rate. But there is will time when nutrient is going to be depleted because the fixed amount of nutrient was provided, and hence they have nutritional stress condition. My all words are important here. Nutritional stress, and in this nutritional stress condition, whatever the phase is there, it will be known as stationary phase. And in that phase, some are growing, some are dying. So hence it will be nearly straight line. And then the bacteria is not getting any nutrient, and hence they will move towards decline or death phase. So remember, in the exponential growth phase, they can produce primary metabolite, and in the stationary phase, they are able to produce secondary metabolites. Now, in the bacterial growth, one important thing is generation time. The generation time of each and every bacteria is differed. Generation time, it means doubling time. So the bacteria they require some time to become double, it is known as generation time. And it is represented in terms of log nt minus n0 by 0.301. And this type of equations also you have solved earlier, like log phases. For bacterial growth, you can start with the log phase. If you want to calculate the generation time of the bacteria, obviously we have to focus on log phase because they are not growing in the lag. So in the log phase, whatever the bacteria you are inoculating in the initial culture, they start growing. And in the first generation, they becomes double. It means one bacteria becomes two. If it will be two, then it will be four. If it will be four, it will be eight. If it is 100, of course, it is 200. So it is increasing two times. Then after first division, in the next generation, it is 2 to 4. In the third generation, 4 to 8. So 1 to 2, 2 to 4, 4 to 8. So the pattern will be like 2 to the power 1, 2, 3, etc. So you will get finally, if you're going so on after n time period, you will have one type of equation that what is the number of bacteria after time period t. Say for example, if I'm saying that I'm going to start with the 100 bacteria, and if I'm providing nutrient properly, this 100 bacteria, in the initial phase, it is fine. Okay, they are not growing. But in the log phase, once you start calculating log phase, and that time, this 100 bacteria becomes 200, then 400, then 800, etc. So suppose I'm giving you the time period of the log phase that you are given with the 1 hour time period, and in 1 hour, how many generations are actually passed, so that how many generations has passed so that bacteria becomes double. So of course, for the doubling of bacteria, if it is taking 1 hour, it means generation time is 1 hour. Okay. Uh, Generation time is doubling time. Everyone of us are actually, most of us are actually fixed in our mind that generation time is 20 minutes. It is 20 minutes is the generation time of E. coli, which is a model bacteria. But it is varying. Like if you are working with the actinomycetes, sometimes it is more than 1 hour, in fact. So generation time is continuously varying with the different bacteria, it is varying. So if I'm talking regarding E. coli, of course, within 20 minutes, it becomes double. So if you start with the 100 bacteria, 100 E. coli, this 100 becomes 200 in first generation, then second generation, 400, in third generation, it is 800. So after 1 hour, 3 generations are passed and they are reaching to the 800 number. So after T time period, that is 1 hour, they becomes 800. And if you're providing where n is number of generation, n0 is initial, that is 100 number bacteria in this example, and nt, that is 800 in this example. So if you are going to solve this equation by taking the log value, it's very simple. Log nt equal to n0 into 2 to the power n, where log n0 into 2 to the power n is basically log m into n, which can be solved as log m plus log n. So log n0 plus log 2 to the power n, and again log 2 to the power n is solved in such a way that it becomes n log 2. And finally, it is coming to the n is number of generation is log nt minus log n0 by log 2. And log 2 is 0.301. This is how you are reaching to number of generation. And you should remember generation time. Generation time is the time taken by the bacteria to become double. And in the terms of equation, it is written as T by N, where T is the time and N is the number of generation. And generation rate is basically N by time, that is number of generation upon time, where time is given in terms of hour here. So you should remember generation time is just opposite of that generation rate. It is inversely proportional. And coming to the next one, that is continuous culture, where you are continuously, you start with a batch, but you are continuously adding nutrient in such a way that it is not going to be decreased. It means nutrition level will be continuously maintained. And the amount of the media you are adding and the amount of media, that same amount of media you have to remove. So addition and removal, you have to balance in such a way that the amount of the nutrient is fixed, the total number of bacteria is fixed, the total product will be fixed. It means amount of nutrient, the number of bacteria, and the product. See, I'm talking regarding three only: media, bacteria, product. If all these three are constant throughout the culture, it is called steady state condition, and that we achieve in the continuous culture. So here, this steady state can be maintained by two way. You have to measure the bacteria, how many bacteria are present. And accordingly, you have to maintain the number of bacteria, and you have to add and remove media in this way only. So you can simply say that dilution you have to do in such a way that the number of microbes should be constant. And for that, either you have to measure the number of bacteria. Bacteria can be measured by turbidostat or chemostat method. In turbidostat, basically we are maintaining the turbidity, and in chemostat, basically we are measuring the limiting substrate, the substrate which must be required by the bacteria for their survival and product formation is known as known as limiting factor. And this limiting factor are main focus area of the chemostat. So chemostat is the most reliable because in turbidostat, you are not knowing that whether bacteria is living or dead. But in chemostat, you can find that bacteria is living or dead. So it is more reliable technique used in the continuous culture. This is just a diagram which is showing that you can add fresh media and remove in such a way that the media becomes constant, the micro becomes constant, the product becomes constant. Then it is called a steady state condition. There are certain specific terminology that you should remember here is the synchronous culture. It means if you start the microbial growth, if all the microbes will be present in the same state, it is called synchronous culture. And next is axenic culture. When you culture the bacteria and you are working with only one pure strain, then it is called axenic culture. And generation time, as I already mentioned, that average generation time is varying between 1 hour to 3 hours, but the bacteria E. coli is having generation time of 20 minutes. Okay. So this is all about the theoretical part. Let us see some questions of this, the last part, that is the culture. Generation time of E. coli is, it's simple, 20 minutes. The organism which obtain their energy from chemicals are designated as. So what will be the answer? The organism which obtain their energy from chemical. It's easy. Is chemotrophs. Lag phase is also known as, so it is period of initial adjustment. It is very easy to solve. And some organisms can use reduced inorganic. Whenever I am saying inorganic, don't see anything. Directly come to the lithotrophs. Okay. So the next question is a starvation condition. Starvation means hunger, nutritional stress condition. If bacteria is having such type of nutritional stress condition, obviously they will be present in the stationary state condition. And if I'm talking regarding generation time, this is very tricky. You can see, and I'm expecting from you that you can solve this. Generation time is, A number is of course correct. Time required for the population to double. Time required for the initial adjustment, no, because it is related to the lag, and obtained by expressing T by N. Yes, I have given this equation, T by N. Generation time, so answer will be both A and C. This question also you can solve if the question is asked in this way, that if a culture start with a 50 cell, then after how many cell will be present after 5 generation? So in the first generation, 50 becomes 100. In the next generation, 100 becomes 200. Third is 2 to 4. Fourth is 4 to 8. And fifth is 8 to 16. So obviously answer will be 16. And during exponential phase, what will be the growth rate? So growth rate is, of course, it will be reciprocal of generation time. This is tricky because I'm talking regarding growth rate, that is GR. This growth rate is opposite of generation time. That I have given an equation. The generation time is T by N and the growth rate is N by T. So these are the things which I wanted to share. Excusing for not giving chance to you for solving questions. The reason is very simple. My aim is not solved by you. My aim is you should know what type of question asked from this type of topics. So basically I mentioned so many questions and that particular topic, and I expecting that you can go through such type of topics, and you will also follow the same pattern of the questions. It becomes easier for you, and you are able to solve all the questions. Have a best of luck for the exam.
Thank you so much, Ravi sir.
Yeah. Welcome, sir.
For such a good elaboration of the important and particularly in context topic JAM. There are so many questions from the bacterial growth like generation time and all type of culture. These are really excellently coverage. Okay. So thank you so much.
Thank you, sir.
Participant, I know it's a hammering because you may continue. So I think the test should be mock test number 1. I will share the link. I will share the link at 3:15. You just take a 10-minute break. 3:20, I will share the mock test link, and you go through the mock test. And the questions is also from physics and chemistry, and there's the 30 questions. Each question